Friends, welcome to Biology Made Easy. Today in this video, we'll discuss about cilia and flagella. Cilia and flagella are usually locomotory structures, and they emerge from the cell, and they are the extension of the cell membrane, which are usually found in case of both prokaryote and eukaryotes, but with a major difference. In case of prokaryotes, the structure is completely different and it is made of protein flagellin. But here in case of eukaryotes that we are going to discuss, here the cilia and flagella consist of three different types of proteins, and the structure is completely different from that of the prokaryotes. So now we will discuss only the eukaryotic cilia and flagella. Sometimes some students confuse whether cilia and flagella are two separate structures or same structure. Actually, both cilia and flagella, they are similar structure. Structural, they are similar. Only the difference lies that the cilia are present in a more number. They are numerous but small in size. They are mostly present in most of the ciliates, while the flagella are very long and only one or two per cell. That is one difference in a structural point of view. There is also a difference from the functional point of view. The cilia, they behave like oars. They behave like oars. As if you have the experience of boating in some lakes, and when we try to move the boat, we push the oars to one side behind, so that the boat moves ahead. We push it. So that is how the cilia behave. That means again they may be isochronic or they may be metachronic. In isochronic, all the cilia that are present, they will function simultaneously. While in metachronic, alternately, 1, 2, 3, 4, like that. So these are the two differences between the cilia and flagella. Structurally, they are same, but cilia are more in number, and the size is small, while flagella are long and less in number. First difference. Second difference is in the function. The cilia, they behave like oars and they push the water behind so that they move ahead. But in the flagella, there is a gliding movement. And how that gliding movement we will discuss only after discussing the structure of cilia and flagella, then we will be able to understand. Now usually in the cilia is surrounded by a cell membrane. That means there is one cell membrane. This is the cell membrane, and within the cell membrane there is a 9+2 arrangement of fibrils. So the 9+2 arrangement is there will be 9 peripheral ones, so you see there are 9 peripheral ones, and these are known as peripheral doublets. These are known as peripheral doublets. Peripheral doublets, and these are made of protein tubulin. In addition to that, there are two tubules at the center, which are connected by means of a bridge. They are connected by means of a bridge between them, and again they are covered by sheath. That means these are the peripheral doublets and these are the central singlets. Then this is known as the sheath and this is known as bridge. This is known as bridge. So here you see there are 9 peripheral doublets, means 9 into 2, 18, and at the center 2. That means 20 microtubules are there, whereas in case of centriole, 9 into 3, 27 microtubules are there. And like that here also the peripheral doublets are made up of tubulin, and the inner one is known as A and the outer one is known as B. Usually the A has 13 protofilaments, while B has only 10 or 11 protofilaments because that is fused with that. Now again the B of one is connected with the A of another by means of linkers, and the linkers are made of protein Nexin. That means see, this A is connected with the B, this A is connected with the B, A with the B. Likewise each A is connected with the B. Like this. So these are known as linkers. And they are made of a protein Nexin. The linkers are made of a protein Nexin. So the first one is tubulin and this is Nexin. These are usually linkers. Then in the inner microtubule, that is A, there are 2 arm-like structures. Here there are 2 arms. This is first arm and the second arm is slightly hook-like structure. So in each one there is again 2 arms are there. One is straight, the other is slightly hooked. So these are known as the arms, and the arms are made of a protein Dynein, and this Dynein has ATPase activity. It has ATPase activity. That means the centriole has a membrane. It has 20 microtubules, that is 9 doublets, means 18, and 2 central ones, and they are made of protein tubulin. Then the B of one is connected with the A of another, that means by linker, that is made of protein Nexin. And in the A of each microtubule there are 2 arms and they are made of a protein Dynein, which functions like a motor because it has ATPase activity. That means there are 3 types of proteins in case of cilia and flagella. And again all of them are connected to the sheath by means of radial spokes. The A of each one is connected to the sheath by means of spokes. So these are known as radial spoke. So this is the transverse section of a centriole. But when we are telling that they are the extension of this cell membrane, in the longitudinal section how it looks like, let us draw one diagram. So in the longitudinal section, if this is the cell membrane, then the cell membrane, it is the extension of the cell membrane. So this is the extension of the cell membrane. And within this cell membrane, this extension of cell membrane is known as shaft, within which there is this 9+2 arrangement structure is there. That will say at the center there will be 2. These are the 2 microtubules at the center, these 2. Then they are surrounded by 9 doublets. So they are the doublets on either side. These are the doublets, and this is known as the basal plate. Now you see, this is known as the sheath. That means the cytoplasmic covering is known as sheath. And this 9+2 arrangement, this is known as axoneme. Axoneme, that there is a 9+2 arrangement. And both sheath and axoneme together is known as shaft. It is known as a shaft. And this is the cell membrane. This is the cell membrane, and this is known as basal plate. Basal plate. Now within the cell membrane there is cytoplasm. Now what happens? Now the two peripheral doublets, they will extend inward. A and B. A and B, they will extend, but the central two will not be extended into the cytoplasm. And in addition to this, in addition to A and B, here there will be also C will be there. The C is not present within the sheath, but it is present here. That we see here, this is A, B, C, this is C, this is B, and this is A. That way 3 are there. And again within the, here there is a hub. This is the hub. This is hub. And at the end, these are known as, from this the rootlets come out. These are the rootlets. And this is the longitudinal section of the, that is cilia and flagella. That means the cell membrane extend outside to give rise to an extension which is known as shaft. This shaft has a sheath within which the 9+2 arrangement structure, that means this structure is present, which is known as axoneme. And then at the membrane of the cell plate is known as basal plate, and then the two peripheral doublets, they enter into the cytoplasm, whereas the two central ones, they do not enter into the cytoplasm. And within cytoplasm, then the third one, that is the C microtubule, is again attached to this. That means this structure is known as basal body, or this is also known as centriole. That means wherever there is a cilia, at the base of the cilia there is a centriole. But wherever there is a cilia, there may not be cilia and flagella. So this is the transverse section, and this is the longitudinal section of this. And almost we have covered the entire structure. That means it has a membrane. It has a membrane made up of 3 proteins, 3 types of proteins, and 3 types of proteins. They are tubulin in the microtubule, then Nexin, it is present in the linker, then Dynein, it is present in the arms. 3 types are there. Then there are 20 microtubules. 20 microtubules are there, and arrangement is 9+2 arrangement. 9+2 arrangement is there, and particularly the Dynein protein, it has 80 page activity. 80 page activity. Now this is the structure of this cilia and flagella. But what is the main function? The main function of the cilia and flagella is locomotion. It is the main function is locomotion. As already I have told you earlier that the cilia, it behaves like oars and pushes the surrounding water so that it moves ahead. But in case of flagella, how it moves, that is the sliding movement. So how that sliding movement takes place, just you see. Now each A microtubule has 2 arms. attached to A, and they are made of protein Dynein, which has ATPase activity. Now they behave like motors, and when this due to ATPase activity, when the arms they either turn either to left or to right, depending upon to which direction they will move. So when they will move either left to right, now you see the A of 1 is attached to the B. So when one starts moving to either left or right, then naturally the B of other one will move. When that will move, then that will again connected with the B of another one. So the second one will influence the third one, fourth one, fifth one. So once it starts moving to one side, all of them will automatically move, just like a train. When the engine starts and it starts moving, all the that is compartments, they move along with it. In the same manner, this is known as sliding movement. When one arm having ATPase activity starts moving, then since all are connected, automatically moves. This is the sliding movement. So that is the difference between the movement of cilia and flagella. And in addition to the locomotion, also in some cases the cilia and flagella, they help in the swimming in water, and in some cases the flagella helps in capturing food, and in some cases it helps in circulation of the food within. Circulation of food. So this is all about the structure and function of the cilia and flagella, and here some important aspects are there. That is 20 microtubules, 3 types of protein, 9+2 arrangement, and how the cilia behave like oars, while the flagella they have sliding movement due to the ATPase activity of the Dynein protein present in arms. I hope you have a very clear understanding. Thank you very much for watching the video. Wish you a very bright future. God bless you.